Hi, this is Dr. Londeville, and this is just a quick little podcast to help you write your hypothesis. Everybody's going to do this in the lab, and you're going to turn it in by February 7th. And this is going to be harder than you think, so give yourself some time to do it, even though you're just writing a few sentences. Okay, so what are the rules? You have to use the language, I will test the hypothesis that. So every single one of these that's handed in, it's going to start with the words, I will test the hypothesis that. It has to be original. You can't copy anybody else's hypothesis. And also hasn't been done before. That's what I mean by original. So it has to be testable within the confines of this lab, meaning that you only have 4 or 5 weeks to do the experiment. You have the equipment that we have. You have a very limited budget, so you can't use anything that's illegal. All kinds of people want to do stuff with illegal drugs. And what I ask you is, where am I going to get the cocaine you're going to use for your experiment? So you can't do anything that's illegal. We can't deal with anything that's infectious. Lots of people want to feed the worm some kind of infectious bacterium or infect them with some kind of virus. We just don't have the capacity to do that safely, so you can't do that. You can't use any equipment outside the lab. So some of you are working in other people's labs, or you have access to a mass spectrometer or, say, a 2-photon microscope. You can't use that equipment for this experiment. You have to use all the stuff that is in the lab only. Okay? Must be interesting and significant. So that means we're going to learn something new about the cell physiology of C. elegans by you doing this experiment, and you're not just confirming something that is obvious or something that has been done before. Okay? You can't make any grammatical errors or spelling errors in your hypothesis. You have to give me some kind of background paragraph. I'm not going to grade the background paragraph, but that tells me about why are you asking this question, what's the context for this question. It helps me judge, is this a good hypothesis or not. And don't have any citations in your hypothesis. That's not appropriate for the sort of thing we're doing. There are occasionally situations where you're testing a particular hypothesis that is famous for some reason, like the blah 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 hypothesis, and you are referring to a paper that explains that hypothesis. But we're not going to get into that sort of thing in this lab, so no citations in your hypothesis. Okay, so example hypothesis. This is a bad example. Feeding worms protein from Impossible Burgers increases their protein concentrations. Why is this bad? Well, it doesn't use the required language. So the rules are, you have to say, I will test the hypothesis that. Okay, does not name the species of the worm. I don't know if you're using C. elegans, or you're using lumbricus, which are earthworms, or there's a lot of things out there in the world that are worms. Does not address a compelling problem. So why do we want to know this? Why is this interesting in a cell physiology class to be seeing whether or not feeding worms protein increases their protein concentration? So this is like actually a really popular sort of hypothesis that I've gotten in the past. I don't know why. I don't know if this comes from people's interest in bodybuilding or that sort of thing. And you should know intuitively that this isn't going to work, because just eating a lot of protein doesn't make you more full of protein. And your body knows how to convert any kind of excess calories into fat. So I don't really understand why this is popular, but it is popular. There's no cell physiology basis to this idea. There's no mechanism. Cell physiology is all about how does this work, and this doesn't get at how it works. Okay, a mechanism, a biological pathway, a gene that's involved, something along those lines, not just general macromolecule causes this effect. Also, you can't do this experiment. So after you do Wormorama Lab, you'll realize, hopefully, that these worms are eating bacteria, and you can't just put like protein from this source on a plate and expect them that they're going to pick it up. It's just not how the biology of those worms work. It's just not how they eat. Along those lines, people will say something along the lines of worms being pregnant, or worms, or some kind of thing that involves the use of hundreds of male worms. After you do that Wormorama Lab, you'll just know that that's not practical, and it doesn't make sense. Or people will talk about the brains of the worms or the liver of the worms, and they don't have livers or brains. So you got to understand the biology of the organism you're working with, and don't say something silly like that. Okay. And then finally, why do I want to know this? What problem is it solving? What is an issue in cell physiology that this solves? It doesn't. So all those things make this a bad sort of hypothesis, not worthy of the time and money that you would spend to pursue this hypothesis. Okay, and there's a spelling mistake. That's not how you spell protein. There's also no background paragraph. Okay, those are rules that are part of the assignment. Okay, here's another bad example that is less obviously bad. I will test the hypothesis, so using the right words, that RNAi feeding that targets AMA1. AMA1 is, in italics, is how you would indicate the gene for this particular thing in C. elegans, reduces expression of AMA1, okay, the gene AMA1. And then here's a paragraph that go along with it. RNA polymerase II is coded for by the AMA1 gene in C. elegans. Using well-established RNAi feeding vectors, I will feed wild-type worms to knock down AMA1 expression and then test for AMA1 protein concentrations in worms before and after knockdown. Okay, this sounds perfectly reasonable, but why it's not great is that we already know the answer to this question. Okay, so it is specific. It is cell physiology based. There's a pathway here. There's a gene involved. It uses techniques that we can do in the lab. We can do knockdown in the lab. We can order feeding vectors. We can test for expression of this gene using the techniques that you're going to learn in lab. All that's good. However, this technology has been done over and over and over again. 40 plus years of this technology exists. So that this is essentially asking, if I knock down this gene, it'll get knocked down. And the answer is yes. So we know that this technology works. This feeding RNAi technology works in that it will knock down this gene. So you don't need to address that in a hypothesis. Now, if you were going to, you're going to see this in a second, if you're going to use this as part of a more involved hypothesis, you want to verify that your gene is actually knocked down. But that's just like a control you're going to do along the way. That's not the hypothesis you're actually going to test. So what I'm trying to get at here is, don't choose something that's obvious. Don't sort of say, I'm going to do a protein assay to see if there's protein there. We know there's protein there, okay? We know that this technology works. Do something more sophisticated than that we don't know the answer to, okay? You're not trying to confirm somebody else's results. You're trying to do something that hasn't been done before, ever, by anybody, ever, on the planet. Okay, here's a better example. I will test the hypothesis that knockdown of AMA1 in C. elegans via RNAi feeding reduces expression of IRE1. Now, notice a couple things here. AMA1 is in italics, IRE1 is not in italics. That's because here I'm talking about the gene, here I'm talking about the protein. Okay? And I'll post some rules about how to indicate different parts of the central dogma if you're talking about the gene, if you're talking about the protein. Okay, and here's my paragraph. RNA polymerase 2 is coded for by AMA1 gene in C. elegans. Using well-established RNA feeding vectors, I will feed wild-type worms to knock down AMA1 expression. So I'm going to do RNAi, and I'm going to use a feeding vector, and all that's good. I'll test for IRE1 protein concentrations in worms before and after knockdown. All that is the same as what I just told you was not good. But here's where I go further. Stress response is activated by the IRE1 system in C. elegans as it is in mammals. IRE1 activates transcription of stable mRNAs of stress proteins. Therefore, not having functional RNA polymerase should not change IRE1 expression. The mRNAs are already made, just need to be activated by stress. If indeed IRE1 expression is affected, it would support the hypothesis that other mRNAs have been transcribed for IRE1 to be activated. Okay, so we'll talk about this in lecture, but the basic idea is that there are these stable mRNAs that animals use to sense stress. And the stable mRNAs are kept in like two pieces. And when stress happens, they come together as one piece. Then they get translated by ribosomes, and they make stress proteins to deal with the stress. Okay? So that's how the system is reported to work. And those mRNAs exist. Okay? But if you don't have RNA polymerase 2, that means that you cannot make any new mRNA. Okay? That's what RNA polymerase 2 does, is it does transcription of mRNAs that turn into protein. So in this system, if I can't make any new mRNA, that shouldn't matter because the mRNA is supposed to be already there, ready to sense stress. But however, if it does matter, if it affects how much of this stuff is made, then that means something else is going on. Some other part of the pathway needs a new mRNA to be transcribed when the animal is under stress. And that would be new. That would be something we don't know yet. Okay? That would be a discovery. Okay? So it puts a twist on the previous hypothesis, and it makes it more interesting. It takes it from something we know to something we don't know. The train of logic is, here's how we think things work now. If that is true, then I can make this other prediction based on that, and then I can test that prediction. Is that new prediction true or false? Okay? So that's how the best hypotheses, or some of the best hypotheses, are chosen, is that there's some kind of twist that's taking something that we know and building on it. Some kind of pathway that we know and asking about it in a new situation. What would happen in this situation, which hasn't been tested before? And so now hopefully you can see that this is writing a one-sentence hypothesis that's good is hard, and it's going to take you a little while. So you're going to have to dig into literature. You're going to have to ask me questions, ask your TA questions, and say, hey, what do you think about this? Can I do this? What about that? Okay, those are all things I want you to do. And each of you are going to hand this in as a PDF by the first week of February on the 7th. This is due. Okay, good luck.